Hi everyone, this video is going to briefly introduce you to some different nutrition research designs that are used, the good, the bad, and the ugly. Nutrition research is very challenging to conduct because of a variety of different reasons. For example, people are really tied to their dietary intakes. There's limitations on how we measure their dietary intakes to make correlations or to make cause and effect, etc. This is really important because as you read research throughout your class to post on discussion boards or looking for assignments, you're going to want to be able to talk about the limitations behind these research studies. There's never going to be a perfect research study, but we do the best we can in research. So how do you decide if you should believe nutrition messages in the media? Oftentimes what happens is any type of headline that you see in the media is largely different than what the actual research shows. So it's really important that you don't just alter your dietary intake based off of an article that was written, or what I like to call mommy bloggers, which are people who don't really cite any references and they're just talking about these fad diets to follow. So some basic questions that you can ask yourself before you make these dietary changes is, when was the research published and was it in a peer-reviewed journal, right? So this means that they followed adequate steps, they have done the scientific method, it's been reviewed by people, and ensuring that they cross their T's, dot their I's. It's basically as solid as they can possibly get it at this time. Was the study done on animals or humans? A lot of the research that we're talking about, especially right now, intermittent fasting, the benefits are found in animals and not humans. And so it's going to be a little bit different in regards to how that translates to humans, since we are not rats. And then even within a human population, what's the diversity? Was it on elderly? Was it on kids? Was it on only females, only males? What was the size of the study? Was it 12 people? Was it 1,200 people? The larger the size, the sample size, the more diversity, the better it is to generalize these recommendations. How long did the study last? Was it a 3-week intervention or was it a 3-year intervention? The longer intervention is going to give a stronger result than that short-term intervention. And then, is it the first time you've heard about it? Wait until there's a consensus. Sometimes research is a little bit behind in regards to finding new things just because you have to give it time in order to get there. So for example, GMOs are on the market and there's just not quite enough research to show one way or the other than they're generally recognized as safe. So here are some common research designs that are used within nutrition. So it can be broken down into observational or experimental. Observation is going to be based off of correlation, where experimental is going to give you a little bit more of a cause and effect. So the 3 types of observational research designs that you might encounter is in epidemiology, case control, and then surveys and questionnaires. Epidemiology is going to look at a particular population of interest. So let's say students at Point Loma Nazarene University. And then they're going to make a correlation between maybe the amount of fruits and vegetables that they consume and compare it to maybe their weight. So it's going to be that one population that they're looking at, making a correlation between their dietary intakes and health outcomes. Case control, you're going to look at 2 different groups. One group is going to have a disease that you're interested in. So let's say a group of people who have type 2 diabetes. And then the second group is going to be the control group, so healthy people. You're going to compare and contrast their dietary habits and determine, well, why did this group of people get type 2 diabetes when this group of people did not? And then maybe you find that the group of people who didn't get type 2 diabetes eat more whole foods. And then lastly is surveys and questionnaires. So surveys and questionnaires are kind of, those papers, we've all taken them, where you're just collecting data and then trying to draw conclusions from it. You want to make sure that you're using a tool or a questionnaire that's been approved by research and is valid though. Clinical trials is when you're actually providing an intervention. So what this means, this is an example here, where you have a group of people, you'll randomly put them into an experiment, which is going to get the intervention, and then the control group, which is going to maintain whatever they were previously doing. This example is looking at vitamin D, and they want to know if vitamin D can cure rickets. So the experimental group is getting vitamin D, where the control group is getting a placebo. Then at the end, they can determine more of a cause and effect because they had an intervention where they compared the experimental group and a control group. Here's a silly little drawing that I like to use to explain a little bit more about the advantages and disadvantages of the research. So you can see the positive is an advantage, the negative is a disadvantage. So again, the epidemiology, I'm looking at a large population, but the disadvantage is that it's just observational. It's not a cause and effect. Animal studies is that the positive is that we can do genetic testing, but this doesn't always translate to humans. The case control, the positive is a correlation, but the negative is you're only looking at that one group that has that diseased state. And then clinical trial, it's a positive that it's a more cause and effect. However, it's not always realistic to translate to real life. For example, in dietary interventions, if they provide all meals in a laboratory, that's not always going to have the same effect because not everybody has that ability to purchase their meals that has the exact micronutrients, exact macronutrients for that necessary outcome. In the end, it's really important to understand that DNA plays a large role in our health outcomes, or genes basically is what that DNA is looking at. So our genetic code does put us at a higher risk of certain disease states based off of our family history. However, nutrition and lifestyle factors have the ability to influence either positively or negatively. So this is one of the biggest limitations to all research is the fact that we each have a unique genetic code that we're not all going to be responding to. So this is something where a lot more research is being plugged into. What I want you to do is read Article 1 and read Article 2 and determine what type of research design was used.